We often talk about our genetic blueprint, the DNA that makes us who we are. But what happens when that blueprint isn't copied perfectly? What if pages get ripped out, or copied twice, or just stuck in the wrong order? Well, that's exactly what we're going to dive into.
Okay, so what could Down syndrome, a specific type of hemophilia, and the way our bodies evolved to carry oxygen possibly have in common? It sounds like a total riddle, right? You've got these three completely different things, but believe it or not, the answer connects everything we're about to cover.
And there it is. All of them trace back to changes in our chromosomes. We're talking everything from tiny little edits to massive, large-scale rearrangements. And this isn't just, textbook stuff. These are incredibly powerful forces that literally shape life, for better and for worse.
So before we can even start to talk about changes, we need to know what the original blueprint is supposed to look like. And this right here, this is a human karyotype. It's the complete master set of all 46 of our chromosomes, all neatly organized into their pairs. Think of it as the master map that geneticists use to find any major deviations from the standard plan. The key thing to get here is that a karyotype gives us that big picture view. Scientists actually take a snapshot of the chromosomes when they're all condensed and super visible. That happens during a phase of cell division called metaphase. Then they can arrange them all in order, from the biggest, chromosome 1, all the way down to the smallest, 22, and then the sex chromosomes. It's this careful arrangement that lets them spot if something's off.
But wait, how do they actually tell all of them apart? Some of them look pretty similar, right? Well, they use a 3-point checklist. First is their overall size. Second is where that little pinched-in part, the centromere, is located. And third, and this is super cool, is the unique banding pattern that shows up when they're stained. It's literally like a unique barcode for every single chromosome.
And that centromere location, it's a huge deal. It's what gives each chromosome its characteristic shape. If the centromere is dead center, you get two arms of equal length. But if it's off to one side, you end up with one short arm, we call that the P arm, and one long Q arm. These different shapes are absolutely critical clues for telling chromosome 3 from chromosome 8, for example.
Okay, so we've got the normal blueprint down. Now for the really interesting part. Let's start digging into what happens when the blueprint itself gets, edited, when there are structural changes happening within the chromosomes themselves. And these edits really come in 4 main flavors. You can have a deletion, where a chunk just goes missing, a duplication, where a section gets copied, an inversion, where a piece gets flipped backwards, and a translocation, where a chunk breaks off and sticks onto a totally different chromosome. So, let's start with deletions. It's pretty much what it sounds like. A piece of the chromosome just vanishes. Poof! Gone. It's like a page gets ripped out of our blueprint. Now, how big of a deal is that? Well, it completely depends on what instructions were on that missing page. A well-known example in humans is Chromosome syndrome, which is caused by a piece missing from the short arm of chromosome 5.
Duplication is the flip side. Here, a segment gets accidentally copied, so you have extra genetic material. It's like someone took a page of the blueprint, photocopied it, and then stuck the copy right back in next to the original. Now, as a general rule, having a little too much genetic info is usually less damaging than having some of it missing, so duplications often have less severe effects than deletions.
But, and this is where things get really cool, duplications aren't always just a mistake. From an evolutionary standpoint, they can be a massive engine for innovation. They are the raw material that nature uses to build something brand new called gene families.
Think about it like this. Once you have a duplicate of a gene, you've got a backup copy. The original gene can just keep on doing its important day-to-day job, but that extra copy, it's now free to change. It can pick up mutations over millions of years without causing any harm. And eventually, it can evolve into a gene with a totally new, but often related, job. We call these related genes paralogs. And here we go, circling back to our opening riddle. The human globin gene family is the perfect example. Way back when, there was one single ancestral globin gene. It got duplicated, then duplicated again, over and over through evolutionary time. Those copies then slowly changed and specialized, giving us the different hemoglobin proteins we need as an embryo, a fetus, and then as an adult. So a genetic mistake ended up being a huge evolutionary win for us.
All right, pencils up, this is a big one for exams. Let's talk inversions. This is when a chunk of a chromosome breaks, flips a full 180 degrees, and then reattaches. Now, there are 2 types you absolutely have to know. It all depends on the centromere. If the flipped part includes the centromere, we call it pericentric. Think perimeter, it goes around the center.
If the flipped part is completely separate from the centromere, it's paracentric. Think para like beside. And circling back to our riddle again, a very severe type of hemophilia is actually caused by a huge inversion on the X chromosome.
Okay, let's look at this visual, because it makes it super clear. Look at B. That's paracentric. See how the centromere is right there, inside the part that got flipped? Now look at C, the paracentric one. The centromere is way over here, totally outside the inverted segment. Seriously, get this difference locked in your brain for test day. It's a classic question.
All right, our last type of structural change is translocation. This is a weird one. A piece of one chromosome breaks off and attaches to a completely different non-homologous chromosome. The analogy here is spot on. Imagine you're reading the electrical blueprints for a house, and suddenly there's a page about plumbing just stuck in the middle. The information isn't gone, but it's in the absolute wrong context, and that can really mess up how genes get regulated.
Okay, let's shift gears. We've been talking about editing the pages within the blueprint. Now, we're going to talk about a different kind of problem. What happens when you have the wrong number of blueprint volumes to begin with?
Okay, another big exam topic here with two critical terms you need to know: euploidy versus aneuploidy. Euploidy refers to having a different number of complete sets of chromosomes. So instead of being diploid, an organism might be triploid, with 3 full sets. Aneuploidy, on the other hand, is a change in the number of just one particular chromosome. This is way more common in us, and it's where you get things like an extra copy of just one chromosome, say chromosome 21.
And within aneuploidy, you have two main situations you'll hear about. Trisomy is when you have one extra chromosome, so that's 2n plus 1, and monosomy is when you're missing one chromosome, that's 2n minus 1. Pretty straightforward.
And this takes us all the way back to the first part of our opening question, Down syndrome. If you look at this table, you'll see it right there. Trisomy 21. That means one extra copy of chromosome 21. And you can see how other trisomies, and even a monosomy like Turner syndrome, lead to other specific conditions.
So you might be wondering, why is just one extra or one missing chromosome such a big deal? It all comes down to something called gene balance. Our cells are like a finely tuned machine. They expect a very specific amount of product, usually a protein, from every single gene. If you have a trisomy, you suddenly have 3 copies of all the genes on that chromosome, so you're making about 150% of the normal amount of those products. That throws the whole system out of balance and can cause major issues during development.
And this is an important point to remember. In humans, the chances of having a child with an aneuploidy, like Down syndrome, go up as a parent gets older. And this link is especially strong with maternal age. The reason is pretty fascinating. A woman's eggs are actually arrested in the middle of cell division from before she's even born. And after decades, the cellular machinery that's supposed to pull the chromosomes apart perfectly can just become a little less reliable. So let's bring it all home. We've seen how these changes in our chromosomes can cause serious, sometimes devastating conditions. But we've also seen how over the vast expanse of evolutionary time, things like duplications have been the creative force behind new genes and new abilities, like how we carry oxygen. It really leaves you with a big question to think about. Are these genetic mistakes really mistakes at all? Or are they the very engine that drives the incredible diversity and complexity of all life on Earth?